here the factors that affects the microbial growth. First is the availability of nutrients. So, ang mga organisms or mga microorganism, they need nutrients to sustain their life. So, a particular na environment, may the appropriate na nutrients that is available. So, those nutrients na serve as energy sources, mga microorganism, and they also serve as source and mga elements such as carbon, oxygen, and hydrogen, and other element. Another thing is the moisture. So if there is a moisture, meaning there is a presence of water. So water is essential for life. So living organisms or mga microorganisms, they require water for carrying out their normal metabolic process. So, there are microorganisms or organisms that dies in environments which containing too little moisture, while others grow in a greater amount of moisture. Every microorganism has an optimum growth temperature. May the mga organism which they are in the minimum growth temperature, which means na they grow below that temperature. But in a maximum growth temperature or above, those microorganisms will die. The temperature ranges from minimum to maximum temperature. So, which organisms grows differ greatly from one microbe to another. So, like I have said earlier na if other maximum growth temperature, the organism is mamamatay. Pero there are some instances na an enzyme nagdedetermine whether ang present na organisms is mag-grow at best in a large temperature and pH. So with enzymes, may niya optimum temperature and pH ranges at which they operate at peak efficiency. So if an enzyme is now operate at peak efficiency, so ito yan na enzyme will be metabolizing and growing at its maximum rate. May the mga microorganism in which may the nira prefer nga temperature. So dito hira mas na-grow. For example is the thermophiles. So they love heat. So they prefer at hot temperature. While mesophiles in the moderate temperature. Psychrophiles is in the cold temperature. And lastly is the psychodoric organism, in which they prefer warmer temperature, but they can endure in the cold temperature. pH determines whether ang isang solution is nabilong ya acidity or alkalinity. Most microorganisms prefer a neutral or slightly alkaline growth medium. So there is a pH of 7.0 to 7.4. But ang mga acidophilic microbes, such as those nga na-live in our stomach, na preferirahin pH nga 2 to 5. May mga organism nga tinatawag na acidophils, meaning na preferira nga tuhan highly acidic environment, such as an fungi. Then may daliwat mga alkaliphils, so na preferira mga alkaline environment. When we say osmotic pressure, this is the pressure that is exerted on the cell membrane by some solutions both inside and outside of the cell. May nakasinisaring na hypertonic, hypotonic, and the isotonic solution. So when we say hypertonic, the outside or the environment of the cell is greater than the concentration of solutes inside the cell. So, if magkamay the loss of water, it causes the cell to shrink. So, for example, ang mga cell membrane ng cytoplasm na shrink tikadtuha cell wall. It is in the hypertonic solution. But when we say hypotonic, ang iya outside the cell is lesser than the concentration of solutes inside the cell. So meaning, kung magkamay the increase of water within the cell, it will cause the cell to swell. Then, if there is an equal concentration of solute outside the cell nganin inside of the cell, the solution is said to be isotonic. Let's proceed to the salinity. When I say salinity, it contains a salt. So there are microorganisms that can survive and even grow ang mga salty environment. Like for example, those mga microorganisms, they prefer salty environments. So they are called the halophilic. While there are organisms na dire na prefer to live in an environment which is salty, but they are capable of surviving. Those organisms are the halodoric organisms. Another factor is the barometric pressure. Most bacteria are not affected by the minor changes in the barometric pressure. So we have some organisms or microorganisms that thrive in the deep ocean and in the oil wells where the atmospheric pressure is high. So they are called the barophils. For example, ang mga archaeans, they are considered as barophils. Why? Because they are capable of living in the deepest part of the ocean where there is a high temperature. And lastly is the gaseous atmosphere. So, nakadepende ha type ng gaseous atmosphere, it require heat o sa microorganisms. So, therefore, ang type ng concentration ng sa gas na present sa environment is not determined whether and microbes are able to live. For example, if an environment is in the laboratory, then it requires a concentration or the increased concentration of a carbon dioxide, which is known as the capnophils. Encouraging the growth of microorganisms in vitro. So here are some sites where the growth of microorganisms are encouraged. So for example, mga clinical microbiology laboratories, itira ginhimo is gina-isolate nila ang mga microorganisms from the clinical specimen, then ira ginpapagrow sa culture media para ira makagather in information na ma-identify nila kung what kind of pathogens is present. While ang mga research laboratories, ang mga scientists is nagko-culture hirahin microbes so they can learn more about them. Then, diba mga antibiotics is tikang hirahan mga microorganisms. Ginproduce sirahan microorganisms. So, from that, nag-harvest sila mga antibiotics and other microbial products para mag-testhin new nga antimicrobial agents nga na-producehin vaccine. And lastly is in the genetic engineering laboratories. So, genetic engineering laboratories, ira ginimo is nag-i-introduce sirahin mga new genes sa cells. Then, once ato na nga new genes is ma-introduce na sa cell ng mga microorganism, ato na mga microorganism is makakag-produce sirahin new gene product. When we say culture media, it is material where any microorganisms may grow and develop. A media that is used sa mga microbiology laboratories to culture bacteria are said to be artificial media. Artificial kay dili na occur naturally, but they are prepared in the laboratory. One way to classify culture media is based on the exact contents of the media. So, kung ang tanan na ingredients are known or ada mismo laboratory, it is said to be chemically defined medium. But, if the contexts are not known, it is a complex medium. Culture media can also be categorized as liquid or solid. When we say liquid media, these are contained in a tubes. While it's solid media, it is prepared by adding agar to a liquid media. So agar is polysaccharide which is obtained from red marine alga. So it is used as solidifying agent. This time, i-differentiate liwat natin ang enriched media, selective media, and differential media. When we say enriched media, it contains a rich supply of special nutrients to promote ang growth ng mga fastidious organism. So, kailangan niyahin extra nutrients, which is the nutrient agar. Well, selective media are used to grow only specific types of a microorganism, while gini-inhibit ang growth of other unwanted microorganisms. And lastly is the differential media. So, ginagamit niya para i-differentiate and different groups of microorganisms that grows in a media. Inoculation of culture media. If ating gagamitan is ang liquid media, what you're gonna do is mag-adla kitang portion ng specimen na to medium. Pero kung solid medium, is kailangan natin gumamitin sterile inoculating loop. Then, ato na sterile inoculating loop is gagamitin natin pagkuhain portion ng specimen, then ikakadto ha surface han medium. It is necessary nga gumamit kitahin sterile technique kay para ma-exclude an all microorganisms sa usang particular nga area so that ito na area is maging sterile. Ngan to avoid unwanted organisms such as ang mga contaminants. So iyagin may minimize ang possibility and contamination. After ma-inoculate ang media, they must be incubated. So, ginpe-place sira sa nga incubator nga na-contain in appropriate atmosphere, moisture, nga appropriate nga temperature. So, may the 3 types in incubator na ginagamit sa mga clinical microbiology laboratory. First is an carbon dioxide incubator. So, ini nga incubator is ginagamit para i-isolate ang mga capnophils. With capnophils, ang mga organism nga na-grow in an atmosphere that contains an increased carbon dioxide. Next is the non-carbon dioxide incubator. An incubator nga na-contain in room air. So, it contains oxygen. There is no carbon dioxide. Then lastly is the anaerobic incubator. This is an incubator which contains an atmosphere devoid of oxygen. Bacterial population growth curve. So, through niya nga population growth curve is any particular species of bacteria can be determined by its growing pure culture of the organisms in a liquid medium at a constant temperature. So, there are phases in the growth curve. So, first phase is the lag phase. So, during this phase, ang bacteria is gina-absorb niyan tanan ng nutrients na ginsynthesize ng mga enzyme para makapag-prepare hand cell division. The second phase is the logarithmic phase. So, during ini nga phase, madagmit ang pag-multiply han bacteria. Ang number ng organism is nga double during each generation time. So, during ini nga phase, ang iya growth rate is hataas. Next is the stationary phase. So, kung ang nutrients ha liquid medium na ginagamit is hataas, then ang concentration ng toxic waste products ha mga metabolizing bacteria is nahataas gihap. So, ang iya rate and division is nahabobo. Then, ini nga phase is ang culture is may daniya hataas na population density. And lastly is the death phase. So, during ini na phase is ang mga microorganisms is namamatay. Only few it nakaka-survive. The reason kung kaya namamatay mga bacteria is because nagkakamay dahil overcrowding kay tungod ng population. Then, ang concentration ng mga toxic waste products is na continue pagataas, while ang nutrient supply is na decrease. Culturing obligate intracellular pathogens in the laboratory. As we know that obligate intracellular pathogens are those microorganisms which can only survive and multiply within the living cells, so they need the host cell. Here are some 3 ways in which the organisms may grow in a laboratory. So they must be inoculated into an embryonated chicken eggs, into the laboratory animals, or either in the cell cultures. Inhibiting the growth of microorganisms in vitro. Here are some sites where microbial growth must be inhibited, such as hospitals, nursing homes, and even mga healthcare institutions. Kailangan niya i-inhibit so that ang mga patient is diri ma-infect, and other staff members or even ang mga visitors. Then, mga food and beverage processing plants, then kitchens, or even mga bathrooms and restaurants. Sterilization, it is the complete destruction of all microorganisms. So, all microorganisms are destroyed, including an cells, spores, and even an viruses. While disinfection it is the removal of pathogens from a non-living object by physical or chemical methods. So meaning not all pathogens are being removed. So may dakitaan pasteurization under disinfection. Pasteurization is a method of disinfecting liquids. For example, mga liquids such as milk and other beverages, ginagamit pasteurization para ma-eliminate ang mga pathogens. Then, ang disinfectants. So, ang disinfectants ang mga chemicals na ginagamit para i-disinfect ang mga non-living object. Then, an antiseptic, o sayang na solution which is used para ma-disinfect an atom skin and other living tissues. And lastly is the sanitization. Sanitization is only the reduction of microbial population. So meaning, iya laging reduce an population ng mga microbe, pero di rin iya gindi-destroy. Then, an microbicidal agents and microbistatic agents. When we say microbicidal agents, iya ginpapatay ang mga microbes, while microbistatic agents is nagamit iyan mga chemicals or drugs para may inhibit ang growth or reproduction han usang microorganism. In terms naman hit bacteria, when we say bactericidal agents, is the same, ginpapatay niya ang mga bacteria. Then, ang bacteriostatic agents are those na gini-inhibit niya ang metabolism ng reproduction han usang bacteria. Lyophilization is known as freeze drying. So, it is the process which combines dehydration and freezing. It is not used for killing microorganisms. Rather, it is used to prevent them from reproducing, and they are stored for future use. Differentiating these terms. So when we say sepsis, it has the presence of a pathogens, while asepsis, there is an absence of pathogens. Aseptic technique is used to eliminate the pathogens. For example, is hand washing. Then the antisepsis is the prevention of any infection. And lastly is the antiseptic technique. So during ini nga technique is nagamit kitahin antiseptics nganhin an aseptic technique.